Good evening, everyone. Uh, Welcome you all to week 7 NPTEL live session. And today we'll be discussing few questions on RNA exports, specifically on about this siRNA, miRNA, and other RNA exports, how this get exported to cytoplasm after getting post transcriptional modification. Okay, and before that, let me introduce myself. This is Subhasmita Mahapatra from IIT Indore. I am a PhD scholar here. Okay, let me know if my screen is not visible to you or if I am not audible to you people. Okay, fine then. Uh, as I have given a brief introduction about today's session, that is about RNA exports. So before going to moving into completely depth into RNA export, how this get exported to cytoplasm, let's talk about little bit about RNA helicase. So everyone knows the helicase means it helps in helicase activity. That means unwinding of the helix, right? So and that happens during the replication process and also the transcription process. Understood? Next thing is, uh, there are few other helpful functions of RNA helicase which we need to know about this. Okay, so today we'll be discussing about RNA helicase in brief. After that we'll move into RNA export. Understood? So I believe that everyone has gone through the lecture videos. So let's start today's session with the first question, that is, which statement is wrong about RNA helicase? It is a DNA dependent antipase. It hydrolyzes ATP. Its substrate is RNA protein complex. Fourth is it has helicase activity. So here it has been asked that which statement is wrong about RNA helicase. So read the question again and try to answer this question. You can write your answer in the chat box. Okay? Then we will discuss after 2 minutes. everyone try to answer this question. If you read the options carefully, then you will find the correct answer. Okay, let's discuss about it. So I see some of you have said A and some of you have said C. So now you got confused between A and C. Are DNA dependent antipase or substrate is RNA protein complex? So let me clear it for you. Here we are talking about RNA helicase, not DNA helicase. So if this is RNA helicase, then it should be RNA dependent, not DNA dependent. Okay, as the name suggests that it is RNA helicase, and it is generally the main function of RNA helicase is to helps in the secondary structure of RNA with a motor function. Okay, so this is basically RNA dependent antipase. And also the substrate is RNA protein complex. This is correct, because when we are talking about RNA helicase, there must be some proteins are also there, and the substrate is RNA protein complex, which will be clearly discussed in your lecture videos. You can go to that lecture video and you will find there. Okay, I'll just brief the things for you. So about RNA helicases, they are RNA dependent antipases or RNA unwindase. Understood? Remember, as we are talking about RNA, so it should be RNA dependent, not DNA dependent. If it is DNA dependent, then it will be called as DNA dependent, not RNA dependent. Fine. Next is hydrolysis ATP. That is ATP dependent motor. ATP full form is adenosine triphosphate. Okay. Hydrolyzation matlab, what is the meaning of hydrolyze? Hydrolyze means, for example, this is A, B. This is a complex. Okay, AB, this is a complex. If it gets broken in presence of water to A plus B. Okay, if this complex get broken to A and B in presence of water, that is hydrolyzation. This process is called hydrolyzation. Condensation is the opposite of hydrolyzation, where two molecules combine to form a new molecule in presence of water. Okay, if I just reverse the arrow, so this process will be called condensation. Fine. And very good example of condensation reaction is your peptide bond formation in case of amino acid chain. Okay, in case of protein, when two amino acids combine, then combine means the peptide bond formation happens, that leads require a condensation reaction. Understood? Next is substrate is double stranded RNA. Okay, for unwinding purpose, there must be some two helix, right? Two chains. For example, this is a single chain of RNA, so there must be other chain of RNA also there, so that they can unwind to each other. Understood? Or RNA protein complex. This RNA helicase is also used to unwind the or separate the RNA protein complexes also. Fine? Now understood? 7 conserved motifs for helicase activity is already there, and N-terminal and C-terminal extensor for substrate specificity. To carry out, to understand the substrate specificity, the N-terminal and C-terminal extensors are there, which has crucial role. We'll be talking about it. Understood? So now you got your answer. Which statement is wrong about RNA helicase? So this must be RNA dependent. Fine? Okay then. Let's move to next question. Which process require RNA helicase? Now we just talked about what is the important features of RNA helicase. Now let me just answer me. Which process require RNA helicase from these 4 options? Glycolysis, nucleotide biosynthesis, ribosome biosynthesis, or none? All these questions are are extracted from your lecture videos only. Okay, try to answer this question. We'll discuss it after 2 minutes. Okay, then let's discuss about it. As I see that most of you have answered C, that is ribosome biosynthesis. So here is it is the correct answer. So this is a figure which shows the processes requiring RNA helices. So you can see here that this is the ribosome biosynthesis process where RNA helices have been required. Also the transcription process is important where RNA helices is required. Ribosomes require assembly of nearly 100 proteins and 4 rRNAs. rRNAs means ribosomal RNAs, okay? And 35S rRNA processed by a series of endo and exonucleases. Okay, so what is the meaning of endonuclease and exonuclease? Endonucleases are those nucleases which break the phosphodiester bond between two nucleotides at a specific sequence inside the nucleotide. Okay, inside the chain of nucleotides. And exonucleases are those nucleases which cut or break the phosphodiester bond at the 3 days end or 5 days end. Those are exonucleases. Fine. More than 14 RNA helicases participate in rRNA processing and ribosome biosynthesis. Now, for ribosome biosynthesis, one essential RNA is rRNA, that is ribosomal RNA. And to process those RNA helicases are required for this process. That is, the number is more than 14. Okay. This is a complex process of ribosome biosynthesis. If you want to go into detail, then you can go for it. Otherwise, it is okay. Okay. Fine. So here is your next question. RNA helicase act as both endonuclease and exonuclease in ribosome biosynthesis. Is it true or false? Read the question and try to answer this. Try to answer this question, everyone. This is a true and false question. As I see, as we have discussed, see, so this is true. Okay, RNA helicases act as both endonuclease and exonuclease in ribosome biosynthesis. It helps in this snRNA and pre-RNA base pairing, activity of endonuclease and exonucleases, intra and inter RNA-RNA interaction. That means intra-RNA means inside the RNA and inter-RNA means between two RNAs. Okay. Why it is required? Because for the processing of ribosomal RNA. Okay. Next is RNA protein interaction. So during the formation of ribosomes, there are different interaction including RNA protein which need to be resolved by RNA helicases. You can go into detail. Okay, understood? Now let's move to next question. Which one is not a function of helicase in RNA transport? Formation of RNA protein complex, unpacking RNA protein complex, or coupling RNA export with translation, coupling replication with transcription. Now we were just discussing about the ribosome biosynthesis. Now we are talking about RNA export. Okay, what is the function of RNA helicase in RNA export? And here you need to click on not. Okay, as I see that you answer D, coupling replication with transcription. So let's discuss about it first. That RNA helicases in RNA export, correct conformation of RNA protein complex for export through NPC. NPC means what? Nuclear pore complex. Okay, this nuclear pore complex is present at the in the nuclear membrane, which is responsible for transporting, exporting or importing any material to and from the nucleus to cytoplasm. Okay, we'll be discussing about nucleoprotein complex. So correct conformation of RNA protein complex for export through NPC. Next, let's talk about why it is saying that RNA protein complex, because we all know that this RNA gets post transcriptional modification, and in that process RNA tailing and capping. In case of mRNA, this tailing and capping process is important, and which happens in the post transcriptional modification. So after this it transported to your cytoplasm, right, for um translation process in the or protein synthesis. But here it is said that RNA protein complex. Sometimes what happens when there is a small RNA molecule is there, so it can get transported with small protein molecules. But in case of rRNA or large nucleotide residue mRNA, they require a large protein and form a RNA protein complex, that is ribonucleoprotein or RNP complex, and that complex then get transported through the NPC. Understood? Next is unpacking the RNA protein complex in the cytoplasm. Because after moving to cytoplasm, it is the role of RNA helicases only to separate the RNA protein complex so that the RNA or mRNA can get through translation process. Fine. Next is removal shuttling proteins for their rapid transport to the nucleus. Okay, shuttling proteins have been removed through rapid transport to the nucleus. Next is your coupling RNA export to translation. It also helps RNA export and translation, because as in case of prokaryotes, what happens, transcription process is coupled with your translation process. Coupled means transcription is happening at the when at the same time translation begins. Okay, this is called coupling. But in case of eukaryotes, we cannot do the same thing, because you know transcription is happening inside the nucleus and translation is happening inside the cytoplasm. So here we can couple the RNA export to translation, and this is the main role of RNA helicages. There are other proteins and other enzymes are available, but we are talking about RNA helicages only. Fine. So coupling RNA export to translation is another role of RNA helicases. Okay, replication with transcription, this is not a function of RNA helicase, and it is not coupled. Replication and transcription, these two things are not coupled. Got it? I think it is clear to everyone what is the function of RNA helicases in RNA exports. So till now we talked about RNA helicase function in ribosome biosynthesis, transcription and RNA export. Okay, next is, okay, try to answer this question. Nucleoporins are located in nuclear membrane, nucleolus, nuclear pore complex or cytoplasm. Which one is it? I think it is quite clear, because it is a conceptual question which is basic, so everyone can answer this. Okay, let's discuss about it then. So you have answered it correctly. Answer is your nuclear pore complex. Fine. So the appearance of nucleus was a central evolutionary transition, but immediate benefits it provided to primordial eukaryote cells remain mysterious. Prokaryotic mechanism of co-transcriptional translation was departed. Okay, this was a prokaryotic mechanism, that is your co-transcriptional and translational. Okay, co-evolution of nuclear pore complexes and nucleocytoplasmic transport machineries allow transport of various molecules. Okay, there are several mechanisms included in this transportation of molecules or things between cytoplasm and nucleus. Okay, and this cytoplasmic transport machineries and nuclear pore complexes are co-evolved for this process in case of eukaryotes. Eukaryotes segregate RNA synthesis and protein synthesis into distinct cellular compartments, and this segregation facilitates regulation of both gene expression and RNA post transcriptional processing. Okay, this thing we have already discussed so many times, so I am not going to repeat it. So if you have any doubt regarding this, then you can ask me. Okay, let's move to next question. During the nuclear import, RAN GTP is present in the nucleus. Is it true or false? As we have been talking about RNA export, and after that the moving things from RNA cytoplasm to nucleoplasm, then it is said that during nuclear import, means when things move to nucleus, RAN GTP is present in the nucleus. Try to answer this. Okay, then let's discuss about it. As I say, most of you have said yes or true. Okay, let's see. So this is the process of nuclear export and nuclear import, and here it is shown as in difference verse sign, or showing the difference between nuclear export and import, where nuclear export signal is NES, and here the nuclear localization signal NLS have been used. Okay. Next is transport receptors or carioferins. They are exportins. In case of export, they are exportins, but in case of imports, they are called importins. It is quite easy to remember, but there are lot of description have been given about carioferins. You can go to deep detail using different reference books. Okay. In the Cooper cell biology, it has been clearly described about carioperins, nuclear pore complex, and this NLS and NES. Okay, you can go into detail using those books. Small GTPase RAN regulates the carioferins. So what are these carioferins? They are basically transport receptors. Okay, and these receptors are called exportins in case of export and importins in case of import. So RAN GDP is present in the cytoplasm in case of export. So here in case of export, you will find this guanosine diphosphate in the cytoplasm, and in case of import, it is GTP, that is guanosine triphosphate is in the nucleus. Fine. So, and next is this RAN GAP, that RAN GTPase activating protein for nuclear export, and RAN GEF, that GDP exchange factor is responsible for nuclear import. Fine. Export in cargo RAN GTP complex in the nucleus. So in case of export, this exportin, that is transport receptor, cargo means those things which you need to export to cytoplasm, for example your RNA or protein. Okay. Next, RAN GTP complex. These three things form a big complex which will transport it to cytoplasm. Understood? And it is in the nucleus. Next is your importin cargo complex in the cytoplasm. Importin, that is your transport receptor. Cargo means what you need to transport from cytoplasm to nucleus. This complex is in the cytoplasm. Okay. Okay, next, this thing, the whole complex is released in the cytoplasm, and this whole complex using GTP will released in the nucleus. So overall expression, you will find that GDP, that is guanosine triphosphate, has played a crucial role in nuclear import and export. Okay, in most of the metabolic processes, you will find ATP. ATP has been used, so that's why it is called energy currents of the cell. Okay, ATP, that is adenosine triphosphate, but in case of nuclear import and export, guanosine triphosphate is used. This is the main you may find it a differentiation between using different other metabolic processes and nuclear transportation. And these all the things have been, this all the points have been briefed about. You can go into detail using the reference book, as I said, and also go into videos for the details. Okay. So here is your next question. Which one is an example of exportin? Now we have just discussed about it. That is, exportin means what? Export factor. The transcription factor, uh, transcription receptor in case of nuclear export is called exportin. So which one is an example of exportin? mRNA, small RNA, ribozyme, or miRNA? Which one is it? Everyone try to answer this. Okay, let's talk about it. So, most of you have said B, small RNA. But here you will find two answers, that is miRNA and small RNA. Okay. In case of this 4 RNA specifically, rRNA, tRNA, snRNA, and miRNA, that is microRNA, involves exportins of the karyofurin family and RAN cycle. And mRNA, it is mechanistically different, require adapters and release factors. So what is the example of it? Can be both, because small RNA is a big family, under this miRNA comes. Okay, but if you see, if the small RNA is not here, you can directly answer miRNA. Okay, miRNA is more, if you talk about B and D, that is small RNA and miRNA is most acceptable as comparison to small RNA. But small RNA is a big family which under which this miRNA comes, and your small that is your snRNA and other RNA comes. Okay. Next is rRNA transports to cytoplasm via large ribonucleoprotein complex. Is it true or false? Okay, so some of you have said true, and it is true. As we have already discussed, that RNA, that is ribosomal RNA, is a big molecule, and also it requires your proteins to transport via large RNP complex, the large ribonucleoprotein complex. The different RNA species exported through the nuclear pore complexes via mobile export receptors. Okay, so these export receptors includes your importins and exportins. Last, as we talked about, small RNAs such as tRNAs and microRNAs follow relatively simple export routes by binding directly to export receptors. Okay, so what is these export receptors? These are exportins. Understood? So this can be tRNAs and microRNAs. All these, they just directly bind to the export receptors. Large RNA molecules such as ribosomal RNA and mRNAs assemble into complex ribonucleoprotein, okay, particles, and recruit their exporters via class specific adapter proteins. So for this rRNA transport and mRNA transport, they require a complex process, but in case of tRNA and microRNA, it is quite simple. Understood? Export of mRNAs is unique as it is extensively coupled to transcription in yeast and splicing in metazoa. So, this mRNA export is coupled to transcription in yeast and splicing. That means mRNA export is happening and splicing is happening in the same at the same time in case of metazoans. Understood? Fine then. So I think it is clear for which RNAs are transported uh, in the ribonucleoprotein complex and which RNA just transport using the exportin or export receptors. They don't require much protein and complex mechanism to transport. They are microRNAs, tRNAs, and like this. Understood? Here is the next question. What are the number of nucleoporins present in the NPC? Now we just talked about the nucleoporins, that these are present in the nucleopore complex. So what are the number of nucleoporins present? You can guess it if you don't know. I'll tell you the answer. Okay. Let's talk about it. So NPC, that is nuclear pore complex, is formed by 30 different nuclear pore proteins. These are nucleoporins that exist in 8 or 16 or sometimes 32 copies per NPC. Okay, per NPC means in case of one nuclear pore complex, there are about 30 nuclear pore proteins, that is nucleoporins, are present. Understood? Nucleoporins are grouped into three major classes. The first class are the Fg nucleoporins, which contain phenylalanine glycine rich repeat. Glycine is a simple amino acid and phenylalanine is a aromatic amino acid. Domains that fill up the active transport channel and function directly in nucleocytoplasmic transport by meditating the passage of soluble transport receptors. Okay, so you don't need to go through details which transporter is there or which is not, but you can just remember that the class of nucleoporins, that is Fg nucleoporins, they contain a phenyl glycine rich repeat, okay, that fill up the active transport channel and function directly in nucleocytoplasmic transport. Understood this much? The second class are nucleoporins that are devoid of Fg repeat sequences. Those do not have Fg repeat sequences. These are the predominant structural constituents of NPC. Fine. Next, third class are NUPS, which are integral membrane proteins and are thought to anchor the NPC in the nuclear membrane. So, which is required to anchor the NPC in the nuclear membrane? That is NOPs. That means they are specifically required to keep this NPC, nuclear pore complex, in the nuclear membrane. Understood? So these are the first class, second class and third class of nucleoporins we have just discussed. Now let's move to your question. Here it is your answer, that is 25 to 30. Understood? Which energy molecule is required for export in T and tRNA complex? We were talking about RNA export, next is nucleopore complex. So here we will be moving towards tRNA exports. Okay, so which energy molecule is required for exportin? Exportin means, you understand, the transport receptor proteins required for export of molecules from nucleus to cytoplasm and tRNA complex. Okay, let's discuss about it. As I see that A and B. So this is your answer. GTP molecule. Okay, guanosine triphosphate. We have already discussed about it. Let's talk about it in detail. Same principle of export in mediated protein export is have been used for tRNA export also, and this tRNA export receptor, that is exportin T, a member of the karyofurin family, binds directly to tRNAs in a RAN GTP dependent manner. As you know, the tRNA are small molecules, so they don't require any nucleoprotein complex to transfer through NPC. That's why they just directly bind to this exportin T and in the RAN GTP dependent manner. The NES, that is nuclear export signal, in the tRNAs are decoded by exportin T. That means those molecules need to be exported, they have this NES, that is nuclear exported signal. Okay, and this NES are not linear motifs but are present in secondary and tertiary structural elements and 5' and 3' terminal. Understood? So they are present basically in the secondary and tertiary structural elements. So in case of tRNA, if you see the structure is like this, in the clover leaf like structure, their secondary structure of RNA is already present. So in that secondary structure of RNA, this NES signal is present. Okay, in 5' and 3' terminal, right? So basically tRNA looks like this. They are okay. So in the 5' and 3' terminal, this might be a 5' reason, and this is your CCA arm, and after completing, so here it will be the NES, that is nuclear export signal, will be present. Once this tRNA export T RAN GTP complex reaches the cytoplasm, RAN GAP stimulates the GTP hydrolysis. GTP means guanosine triphosphate become hydrolyzed to on RAN to induce release of tRNA cargo. Exportin 5, another member of karyofurin family, might be an auxiliary receptor. Okay, so this exportin T is an important receptor which helps in transport of tRNA molecule from nucleus to cytoplasm, and there are several other molecules are present which is responsible for this. Okay, here is your next question. Which one is tRNA export receptor? We are just talking about the eukaryotic tRNA export, and in case of this is tRNA export receptor, which one is it? LOS1, ARC1, CEX1 or non? In case of earlier, it is exportin T. Okay, now what is it? Okay, so most of you have said A, that is LOS1, and it is the correct answer. Let's talk about it. Alternate pathway of tRNA export. Each tRNA export receptor LOS1, an ortholog of exportin T, is not essential for viability. Function of LOS1 tRNA export receptor became essential when the activity of an amino acid tRNA synthesis factor ARC1 is impaired. Okay. Next is CEX1 was suggested to deliver the amino acylated tRNAs from nuclear export receptor at the cytoplasmic side of the NPC. Okay. So, what happened? This exportin T molecules were used for this tRNA export, but suddenly they this ARC they are not required for essence or essential for viability, and the loss of function of ARC1 was impaired, and after that LOS1 was found, and in that case tRNA export receptor LOS1 was used for this process, and CEX1 was another receptor which delivered the amino acylated tRNAs from the nuclear export to cytoplasmic side. Next is miRNAs are exported by which karyopheren? Earlier we have been talking about tRNA export. So miRNAs are exported by which karyopheren? Export in 1, export in 5, export in 10, or none? Fine, then let's see. miRNAs are class of non-coding RNAs are exported by the karyopheren exportin 5. miRNAs, that is microRNAs, are produced as larger stem loop precursor in nucleus which mature into single-stranded RNA species that induce post-transcriptional gene silencing. miRNAs encoding genes can be transcribed by either polymerase II or polymerase III. The primary transcript derived from polymerase II, that is pre-miRNA, transiently receives a 5 days cap and a poly A tail. The cap and poly A tail are removed during subsequent miRNA processing. Okay, so this is basically a class of non-coding RNAs, and they are produced as large stem loop precursor in the nucleus which mature into single-stranded RNA species that includes post-transcriptional gene silencing. Fine. They are mostly used for RNA interference also. Specifically, these non-coding gene sequences are used in gene silencing process in different organisms, including plants, and mostly used in genetic engineering. Okay, that is expectant 5. Now here these two are few basic questions. RNA undergoes decay because it lacks coding sequence, it has stop codon, it has intron, or it lacks essential stability features. Which one is it? Okay, as most of you say D, that it lacks essential stability features, so it is correct. It lacks essential stability features. So, which essential stability feature it lacks? Basically, what happens in case of ribonucleic acid, there is a OH group, that is a hydroxy group present in the 2 days position. Okay, and this hydroxyl group, that is OH group, this is highly reactive. Okay, because of this reactivity nature, it can act as a catalyst in case of ribozymes, and also this is also negative impact in case of in for the stability also, because it reacts and it mutates suddenly. Because of this, it lacks the stability feature, so that RNA undergoes decay. Okay, this is the last question of today's session. So length of polyadenile chain or length of poly A chain on mRNA indicates that mRNA is undergoing decay, that mRNA is ready for translation, age of mRNA, or nuclear location. Okay, so it says the age of mRNA. Okay, what is the age of mRNA? So today we have discussed about RNA export, including your miRNA, tRNA, which are small RNA molecules, and they get transported to cytoplasm using only the transport exporters, that is your exportins, and they don't require any large complex mechanism for the transport process. But in case of rRNA, that is ribosomal RNA and mRNA, they require large protein complex, that is ribonucleoprotein complex, to transfer through the NPC. to cytoplasm, and a specific molecule, that is GTP, guanosine triphosphate, is required in also with the RAN cycle for this export and import process through the NPC. Okay, and earlier we have been discussing about RNA helicase, that what is the function of RNA helicase except unwinding process, and for that we got to know that RNA helicase has crucial role in ribosome biosynthesis, that is in rRNA processing, also in transcription process, and also in RNA export. Okay, we talked about it in briefly, so you can go to the lecture videos to understand it more and more. Also you can go for referral books also, so that you can have a better idea about carioferins, NEL, that is a nuclear export signal and import signal. Okay, these are quite good topic to understand. You can go for that and give it a read. Okay, you can ask your doubts regarding this session or anything else to or about this session in next class also and today's session also. Okay, thank you for joining. We'll meet on next Monday. Okay, thank you.